Okay, and also we will switch to English.
Yes.
Because we are foreign collaborators. So we have a vegetation session this afternoon, and we'll start with Philippe Peylin, a general presentation of our Orchidee land surface model. So Philippe, you have like 25 minutes. We'll leave a little space for questions. Look at me and we'll inform you when you have 2 minutes.
Tu me dis quand j'ai 15 minutes, quand je fais 15 minutes?
5 minutes?
Ouais, tu me dis à 15 minutes.
We are a bit in advance, so you can take your time.
Donc c'est là tout Philippe.
I know.
Good morning. Good afternoon.
En fait, au début, il ne met pas bien.
Ouais.
Quand je fais une deuxième fois, il m'affiche l'écran.
Ah, d'accord.
Une fois qu'on le sait.
Okay, so I will try to present our so-called global land surface model, Orchidee. So I'm talking really in the name of a whole Orchidee project team, and you will see the face at the end. So this is a tool that is really central for our team, Mosaic and another team, BioGeo, and it's a tool that is a research tool, especially used a lot for the climate projection. So it's the land surface component of the IPSL Earth system model. But it's also a tool that is more and more used for studying processes, studying photosynthesis, like Fabienne is doing also, or studying adaptation to climate of certain ecosystems, or mitigation aspects. And it's even more of a tool for studying bioenergy, also what is feasible in terms of bioenergy. And it can be run globally, which is in its initial purpose, but it has no real scale. It can be run locally, and we are running it even more locally here in the team to test processes. So how does the model work? Okay. So basically, initially it was designed to solve the energy and water budget. And then later, the carbon cycle has been added, and now a more nutrient cycle like nitrogen and phosphorus. I will come back to that a bit later. So usually this presentation is more oriented toward climate modeling, so more large-scale application. But typically, we divide the Earth system in different so-called plant functional types. So it's a bit of a big word when you're speaking to ecologists, but to things that even if it's a generic concept, you can refine a plant functional type and adapt it to a given species or for a species or whatever. So it's a generic concept, and all grid cells or application can mix a set of plant functional type, and for each of them, you resolve the energy budget. I won't discuss too much, but all the radiation, you calculate surface temperature and temperature in the soil. You resolve the hydrology, I will show a bit more later, and the carbon cycle, but there will be also several presentations on that. As I said, so the hydrology, it's important, maybe it won't be discussed later, but all is centered for the functioning of the ecosystem with the water availability. So now I'm trying to think, given that I've seen what BIAM is doing, what could be of interest for you? So basically, we have problems to calculate infiltration in the soil. So we resolve the different terms of the bare soil evaporation, the transpiration, interception loss on the forest, how much water is intercepted by the leaves. And we also have the snow. So we take into account, we have a snow model with multiple vertical layers. We calculate the runoff, which is the most difficult, or probably one of the most difficult parts, and then the drainage in the soil and the root extraction. So all that is calculated with Richard's equation, advection diffusion equations along 11 soil layer, but all that is flexible. So I'm seeing things where we can collaborate on how plant structures the soil and may help preferential pathway for infiltration of water, how this interaction at the soil surface between the hydrology and the plant is really critical. Of course, this is linked to something that's probably less of interest for you, with all the large-scale transport of water, lateral flow with different water reservoirs to help us to calculate irrigation, flooding plains, and so on. So you have to know that this exists, and we are developing that. For the photosynthesis, I skipped that because I think there is a presentation just right after, in the afternoon, focusing on photosynthesis. Just the big word, Farquhar model, you will know all of that a bit later. If I come back, so for the above ground part, what is a bit new in this model? So as I said, we calculate the light interception, so let's say all the photosynthesis. And after that, the carbon that is assimilated, we have to build some plants. And one thing for the forest, for the above ground parts, initially all these big model, large scale big leaf model were working as just one big leaf and no real demography of the forest. We have included, following a bit the trend, the first of the model that did that was the ED model developed in the US by Moorcroft. And basically we include diameter and age classes. So a plant functional type we have different plant functional type for different ages. We have a diameter classes and we have an allocation scheme of carbon to grow the plant. So the height of the forest is not prescribed. We put seedlings in the model and they develop and they grow to a certain height, and we have to calibrate so that this height is realistic. We have a recruitment modules and we can calculate a whole stand structure with the way it intercepts the light. So this is really to represent a bit the landscape heterogeneity. I will come back to some of these processes. One important thing that we have included in this model recently is to escape from the mortality process of the plant that was usually based as a decay rate, just a constant rate times the mass of carbon in the different pool. Of course, we have, I should have mentioned, we have different pool of carbons for the above ground part, the leaves, trunk, and 8 pools above ground pool. But now we are trying to calculate explicit mortality to wind flow through one example of the biotic effect, the bark beetles, and the fire. We have a fire model that is included in Orchidee. Of course, merging all that is critical because what we do see, for example, in Europe, there's an excess, a decrease of tree canopy in the Eastern Europe, which is basically a combination of different effects of mortality. So the cascade of mortality is important, and we try to represent that in our model. So this is in progress. We are, for the above ground park, also dealing with not only forests, but for crops. So if we look on the left, you probably all know the sticks model. So it's an effort that started a long time ago, maybe more than 10 years ago, Nicolas, you can correct me, or 15. A long time ago. So basically, we've seen, okay, there is the INRAE at Avignon at that time only was developing this sticks model for agricultural, for crops ecosystem. So we started a coupling between the two models, and it ended up after several tries to basically being, we took some of their module, especially for the phenology, the allocation, and the management aspects that we incorporated into the Orchidee model. Of course, the story is not all that great because it leads to a divergence of a branch, and now we are converging back, but it's all the difficulties of maintaining a collective tool, which is on the size of Orchidee. We did the same for pasture, with the so-called PASSIM model that was also at that time developed at Clermont-Ferrand, not developed at Clermont-Ferrand, but maintained and used at Clermont-Ferrand, where we can have animals density on the prescribed animal density, and we can have different managements of pasture, whether it's grazing or cutting. So there's a strong ongoing effort to merge that into our trunk model that include today also, I should have mentioned the nitrogen cycle, but maybe you mentioned the nitrogen cycle. Maybe. So now that we have basically I presented you briefly the above ground part of this vegetation model. We also have a lot of dynamics around the soil carbon dynamics, soil organic carbon dynamics. And so basically it's based on a model which is called Century, which is well known. It's was developed also a long time ago, including the nitrogen and the carbon and the nitrogen cycle. But we have discretized soil, so basically we can now compute for each soil layer the mineralization depending on the water and temperature of the layers. And that is very important to account for things like the priming effect. I don't know if you are knowing that. So basically, the more fresh carbon you give, you have more living bacteria that can develop and further decompose your organic matter that is more linked to the structure of the soil. So there is a lot of things like that. And more recently, some people focus on the soil microbial biomass. So this is maybe, I don't know, something you are working on. So it's an old slide, but at that time there was a test with the Millennia model where you account directly for pools of microbial biomass pool, while this is the Century type of model where we have a slow, fast, and pools and medium term time of turnover pools. And here we have other pools of microbial biomass. And there is even more a new concept with the mimic model. I don't know, maybe Daniel Gold will present that later on. So a lot of development on the soil dynamics. And I want to mention, we have also a lot of efforts to work on permafrost regions, because there is key challenges of how this permafrost will evolve. And there is a strong coupling in these regions between the dynamic of the organic matter, the thermal properties of the soil, and the hydrological properties of the soil, also how it shapes the infiltration and so that's one part. Nitrogen cycle, it's also a long story, and it has been initially developed in a branch of Orchidee called OCN, and then reinserted by Nicolas quite a long time ago now, more than 10 years. No, not yet, 5 years, 6 years. Sorry. So the nitrogen cycle, basically, probably at your scale at the MIAM, it's here we account more for key processes that impact our carbon cycle. And you may have a lot to bring to us in terms of the dynamic of the nitrogen, from nitrogen deposition, nitrogen fertilization, how it in fact affects plants' physiology and soil organic matter. That was an example, not only to speak about processes, but of a recent PhD with Nicolas, where you can see, just take one example over the historical period, the respective contribution of CO2 fertilization versus nitrogen deposition for the extra carbon accumulated by these ecosystems. So we accumulate ecosystem. Still, the CO2 fertilization with this model seems to dominate, but the nitrogen deposition does not play a negligible role. And in green, it's the synergies between these two effects. So this is a tool where you can study this type of combination of effects. Of course, when you start to think about nutrients, everyone says, well, but nitrogen is not the only nutrient. So there has been some efforts to include the phosphorus cycle and its impact on the carbon cycle. And Daniel Gold has developed a branch of Orchidee, it's called CNP, that accounts for both nitrogen and phosphorus nutrient limitation. Phosphorus is more important over the tropical regions than the northern or high latitude regions. And there is a strong ongoing efforts to include that branch back into the main version of Orchidee, because there are key, detailed points that makes this version with the phosphorus a bit more difficult to handle, because you have to conserve mass of phosphorus, and it poses some problem. So that's for the nutrient parts where we are, but probably in the next nutrients, I don't know which one is on the table, but maybe there are some. We also have people that are working on the chemistry. So if you're interested, I mentioned also Juliette here, Latière, because she's more responsible of the chemistry. So emissions of volatile organic compounds. And we also have a slide after on the methanes and the N2O. So there is a group of people really that works either on the impact of the atmosphere of ozone on the functioning of the vegetation and how it can lead to emission of isoprene or different species. So we have a model that is coupled to the photosynthesis and the whole canopy model to calculate chemical species or reactive species compared to CO2 and compared to CO2. There is a big effort, because this is important for climate, around methane, because mitigation is probably going to be also largely focused on methane to reduce our emissions of methane. And the natural emissions of methane due to peatlands and wetlands is large. And we have a model, I would say it's a process-based model, which comes from the initial Walter model that really accounts for production of methane, storage in these organic, very wet soil, and then how it can bubble and escape to the atmosphere. So if you're interested in that, we have a contact here. Elodie Salmon is working on that. Closer to your basically your work, and so it's two slides that Cédric and Fabienne have provided to me. So we are trying to use solar-induced fluorescence to improve photosynthesis. I don't think you discussed that too much in yours. So basically, I don't think I should explain what is solar-induced fluorescence to you, but from these tiny reflectance in the 690 or 740 nanometer band, we can measure that, and we hope to use these measurements to constrain photosynthesis. So that was an approach that led to developments in the Orchidee model of a process-based. I don't like to say process-based. I think we should be very modest when we say process-based, because for you, it's not process-based. For a molecular biologist, process-based is really when you look at the reactions. And for us, we call it process-based. Why? Because we are in opposition to people that develop similar model and that we call statistical model, where they try to have more statistical relationship from experiments that you made here. We try to describe what the photons are doing, not in a mechanistic way, but more our level of process. So we implemented this process-based shift model in the model, and then we said, well, we will use spaceborne solar-induced fluorescence data here. In this case, the OCO2, but there are more tropony instruments now have even better data for the fluorescence. And what we did, we assimilated. By assimilation, we corrected, and we have a whole program which is called data assimilation associated to Orchidee, that can calibrate the parameter of our model with respect to certain observations. And we assimilated this fluorescence data, and this is the OCO2 measurements, and we see that the a priori Orchidee model has too much fluorescence, and a posteriori we reduce that, which led to also, because we were controlling parameters, we were optimizing parameters controlling the gross primary production, the photosynthesis. We had a reduction of photosynthesis that is a bit more in agreement with the data-driven, with other independent estimates. So which was a good aspect or good point of this calibration. So calibration for us not only means having one unique parameters, because we are more and more interested in having uncertainty. Because with this model, the parametric uncertainty is key, and especially for climate projections. And that's central for the next IPCC climate projections, to have uncertainty from the parameter used in our model. Another little approach that I mentioned, I pick up few, which is a new hydraulic architecture. So before that, what all models are doing, they relate soil water to stomatal conductance through a so-called water stress function, which is really empirical and statistical. Basically, zero stress when there is full soil water, and then full stress when there is no water or when we are at the wilting point. And now we escape from that very empirical relationships to involve an hydraulic architecture where the stem of the plant can store water, so the trees can store water. All that, so this is capacitance. It's basically modeled like an electric circuit, and we have resistances for the water transfer from the soil to the potential of the leaf. And basically, I won't go. We were quite happy that with this first implementation, it gives similar result with this very statistical, which is a linear relation between soil water stress and the impact on the stomatal conductance, with something that is now much more process-based for us with some dependency. We could reproduce roughly the same result than we have before. Opening a new area for us is to calculate mortality through cavitations, to validate all these functions with measurements of the potential in the plant. So it's opened a lot of things for us, and even for dry drought event, it has a better features than the previous formulation. Last but not least, because I like it, sorry, I put it because we further develop it, which is a vertical multi-layer scheme. Basically, we are more and more asked to link climate studies with also biodiversity. And so there is a nexus between food productions, climate aspects, mitigations or adaptations, and also preservation of biodiversity. For forest, what we are doing here, so we start to calculate an energy budget for multiple levels in the canopy. So if you imagine your forest is closed, you may have within canopy climate that is different than above, which is very important if you look at questions links to biodiversity, below ground plants, how they can develop. So this is coupled with a full atmospheric turbulence scheme. So we start to have an atmospheric turbulence scheme within the canopy, and we calculate the temperature and the exchange of water and carbon for each layer. And what that does help us, for one site in Germany, Rheinisch, where you can measure between the top of the canopy and the ground a difference of temperature, not much, 1-2 degrees. This is the observations. This is this multi-layer. We go in the right directions, not always having the right profile, but that means we can start to simulate the climate within the canopy. And I think that's very important. That opens possibilities to study processes at the plant level, where you have at each layer of the canopy a different VPD, a different light. For now, we have a discretization of light, but we don't have the VPD and the temperature. And this is the evolution in red of this over a year of these differences between top canopy, below ground, between the observation and the model. This is the first start, but we're happy with. I'm nearly done in 2 slides. And we have a lot of other developments. I first already apologize for all the developments that I haven't shown. We are doing a lot of things on the biophysics. We have a lake model that is included. We are working on irrigations, thermokarst lake, the lateral transfer of different form of organic carbon or inorganic carbon. We develop new plant functional type like mosses, lichen, and shrubs for boreal PFTs. There is plant traits, but I guess, Nicolas, you will mention that because Nicolas Vieuvi is our pushing towards the use of plant traits and coordination between plant traits. We also had some attempt on herbivory, and these I just mentioned before. So there's a lot of things. You have it's a big ecosystem, and in the trees of the how we pilot. So we usually meet every week. We are a group of 15 around people that use this model and co-develop this model, which has been labelized by CNRS as a tool. And in the red square, this is the people from LSCE. So I'm just saying we are quite numerous at LSCE to co-develop this tool. So there's right places to start collaboration. But we are not only, it's not an LSCE model, it's an IPSL collaborative community tool. I'm ending.
Thank you. We have time for maybe one question in the room and one online. So people online, you can push on the raise your hand button so that we can see you. Yeah.
So going back to the plotting fluorescence measurement, do you integrate the fetch of photons, or is it more information of the concentration of frequency?
Maybe Fabian, you want to I think you will reply better than I will.
Okay, so I'm not sure I got your question. So we compute the radiation that is absorbed by the canopy, and then we compute the emission of SIF for each leaf layer, and then we compute the quantity of photon escaping in the nadir direction.
Yes, but to integrate the fetch in terms of photosynthetic yield versus non-photochemical quenching, for instance.
Yeah, so we have a model for non-photochemical quenching to close the budget of photons. So and this is a research field, how to model this NPQ effects.
And there is even more now. There were some recent papers about the structure of the canopy, how it impacts these different vegetation indices. So this is why having a description of the forest and its structure with gaps and so on will help us also to account for that.
Yes, so the transfer model of the usual fluorescence and the leaf the secure, the leaf distribution, the quality. So is there a question online or another in the room? Laurent?
How do you take into account the different species? Because I guess, for example, for a forest, the trees are very different from tropical forest in terms of amount of leaves, amount of photosynthesis.
So basically, we have so-called plant functional types. So far, we have 15. So of course, we separate between grasses. The cropland is a specific world where we can have different crop species. And for the forest, we have temperate, tropical, evergreen, needle leaf. We separate between needle leaf, broadleaf, between evergreen and red green. And then, of course, you say, well, it's very generic, but it's not the same between an oak and a beech forest. But you can, all that can be also used because if you can set an ensemble of parameters for a given species, you can run with many more species. There was a study over Europe with Orchidee, was probably 50 plant functional type corresponding to different groups of species. So it depends on how much information you can inform the different processes or the different parameters responsible for phenology allocation. So it's flexible in that sense.